Aloha, everybody. My name is Isabelle Eiser, and this semester I worked with Dr. Olivia Nigro on our project determining genomic differences among the virulent pathogen Vibrio vulnificus in the alloy canal. So this image over here is a square grid plate showing some of the bacteria that we isolated from the alloy canal. So a little bit of background on Vibrio vulnificus. This is a virulent human pathogen that resides in warm, brackish coastal marine waters, and its residence is generally associated with temperature and salinity. So this upper left-hand graph is showing levels of Vibrio vulnificus that change in relation to seasons. This was completed in the Gulf Coast, and it shows that as we go into cooler months, the levels of Vibrio vulnificus decrease compared to warmer months. And the graph over here on the right, this is a bubble plot showing the relation between temperature and salinity. So Vibrio vulnificus really resides in this salinity of 5 to 15. For reference, freshwater has a salinity of 0 and ocean water has a salinity around 35. So this bubble plot is showing that as the salinity is lower and a higher temperature, there is a lot more Vibrio vulnificus, but there's a lot of variation even within those temperature and salinity range on any given day. So the diameter of the bubble is related to CFUs per milliliter. There are two subtypes of Vibrio vulnificus. There is a clinical type and an environmental type. The clinical type is more closely associated with pathogenesis in humans, particularly gastroenteritis and cellulitis, both of which can lead to sepsis and death. So research in Hawaii on Vibrio vulnificus is pretty limited. Most of it is focusing on the Alawai Canal due to its close proximity to Waikiki, which is a popular tourist destination. However, other research has noted its presence in other brackish waters around Hawaii, which is important because brackish water, specifically in Hawaii, is really big for tourist destinations and tourist activity, even resident activity. So we really need to be looking into those areas. So for this project, we sampled at the Canal site and the sampling site indicated by these two red stars on this graph. So we sampled at these two locations, and then we filtered those water samples through 0.45 micron GN6 filters at 3 different dilutions in duplicate. So our dilutions were 0.1 milliliter, 1 milliliter, and 10 milliliter of water sample. And we took those filters and we placed them on both chrome agar and TCBS media plates. And we grew those plates overnight. And any colony that was blue on the chrome agar and green on the TCBS media, we then streaked onto these square grid plates shown at the bottom. And so we took any blue colony and we streaked it on both plates, and we took any green colony and we streaked it on both plates. And then we let these plates grow overnight, and anything that was blue and green we then selected for further isolation. And this is because the blue on chrome vibrio or chrome agar and the green on TCBS correlates with Vibrio vulnificus. So then we streaked them from further isolation, alternating chrome vibrio and TCBS until we got these really nice isolated colonies. And we did this 3 times. And then we took these isolated colonies and we put them in slant tubes and we streaked them, let them rest overnight and grow. And then we're now waiting to confirm their identity as Vibrio vulnificus with the VVH gene, which is specific to Vibrio vulnificus. So we got 2 sets of results from this experiment. The first was in relation to how correct was the first colony that we pulled from our filter plates. And what we found was if we pulled a blue colony from chrome agar first for further identification using the 2-plate method, we were more likely to get Vibrio vulnificus than if we pulled a green colony from TCBS. And so if we had pulled a green colony from our filter plate on TCBS, we only got a Vibrio vulnificus color matching in the 2-plate method less than 8% of the time, compared to about 25 to 28% of the time when using chrome vibrio or chrome agar. So then we looked at how much Vibrio vulnificus we found at the 2 different locations. And when sampling the canal, we had a larger amount of Vibrio vulnificus per 100 milliliters in the canal versus the harbor. But this was pretty evident even before we did any type of calculation. As you can see in these images on the right, these are our filter plates from the canal sample and the harbor sample at each dilution. And at the canal sample, our 0.1 milliliter dilution, I believe this is either 45 or 60 colonies compared to the 5 colonies on our harbor sample plate. So it was pretty evident from the beginning that there was a difference in the amount of bacteria at both locations. So we then compared our data to historic data. So for both of these graphs, anything black is the historic data, anything red is the canal data, and anything green is the harbor data. And so both graphs are log CFUs per milliliter on CV. And this first graph is comparing the salinity versus the colony forming units. And so our Canal data, we sampled, that was 7 ppt, and it fits in pretty well with the historic data in this curve. However, our Harbor sample at 12 ppt doesn't reach anywhere close to what historic data had. And then when we looked at temperature, both sample sites were 25 degrees Celsius in the water, and there wasn't a whole lot of correlation to the historic data in this graph. And according to the literature, there's like a slightly negative correlation between temperature and CFUs per milliliter of Vibrio vulnificus. But in the Alawai Canal, it's a little bit different because when groundwater seeps in and lowers the temperature, it also lowers the salinity, which can raise the levels of Vibrio vulnificus. So our data just really doesn't fit in very well with historic data in the Alawai Canal. Overall, we found that differing levels of Vibrio vulnificus were noted at both locations, and the current levels didn't reach that of historic data, but there were salinity depressions. And this definitely could have been due to the fact that we sampled on a very, very rainy day, and that could have changed the residence of the bacteria in the Alawai Canal. It could have flushed out a lot of bacteria that was present. So future directions for this project, we want to do more sampling at different locations around Oahu and continue the process of identification, as well as subjecting these bacteria to different environmental conditions and doing full genome sequence analysis. And this research is very important, especially now with the pandemic. We should be monitoring environmental pathogens very closely just to determine their levels and to predict any risks to residents in Hawaii and tourism in Hawaii. So these are my acknowledgements. This semester I was given funding by INBRI. Thank you.